Hello, listeners. What is up? My name is Al Bears with the U.S. Fish and Wildlife Service Southwest Region in Albuquerque, and you are listening to Shop Talk. Today, we are talking shop with conservation law enforcement experts about how they use forensic science to catch wildlife poachers. A few miles off the interstate in the desert west of Albuquerque, there's an acrid fragrance rising among the prickly pear, cane cholla, and juniper plants. There, two large animal carcasses surrounded by a chain link fence ripen under the July sun. Dozens of game wardens wearing uniforms representing tribal law enforcement groups from across the West bear the stench with humor as they review training materials. For today's story, we're talking about how the study of insects and decomposition stages in forensic cases can lead to the arrest of criminal poachers. This is not the most glorious part of a game warden's job, as it means examining rotting animal carcasses and collecting samples of maggots to determine time of death. In July, the U.S. Fish and Wildlife Service Southwest Region hosted training for dozens of tribal game wardens from reservations from across the West. The region's Native American liaison and former federal wildlife officer Joseph Early coordinated the training event funded by the Bureau of Indian Affairs.
Quite a variety of tribes. We have about 36 officers from around the Southwest. We do have a few from California, Hoopa Valley, tribe out here, Standing Rock, South Dakota, and then Rocky Boys, Montana. The rest are a lot of Pueblo tribes and Apache tribes from here in New Mexico and Arizona. It's good for the tribes because a lot of tribes don't get this kind of training to begin with. A lot of these officers typically go through your basic police or BIA academy. They're taught to be street cops, and then they're put out in the field with maybe little to no knowledge. Unless they're raised on the reservation themselves, they know how to hunt, fish, that kind of stuff, but they're really not given the tools or the training to go out and say, investigate a wildlife kill or investigate a crime scene that is involving, a poached animal, things of that nature. So they get the training when they can. So I work closely with BIA, who provided funding for this training, as well as with the what's called the Native American Fish and Wildlife Society to put this 40-hour training on for our tribal officers.
A major theme of law enforcement for game wardens is the investigation of animals taken illegally for trophies. Often these laws are enforced by the Lacey Act of 1900, which prohibits the trade of wildlife that were taken illegally. Another is the Migratory Bird Treaty Act of 1918 and the Bald and Golden Eagle Protection Act from 1940.
Particularly here in the Southwest, a lot of times you get folks coming in for big game trophy hunts. For example, folks that are head hunters looking for large antler bucks, deer, bighorn sheep, where the case might be. So they're out there poaching animals. So you might come across a carcass with a missing head, and they have to investigate, how the animal was taken, who took it, things of that nature, where they might partner then with U.S. Fish and Wildlife law enforcement if it involves something like a Lacey Act violation. Migratory birds, for example, are a big thing. People are shooting hawks, eagles for feathers, which they use for tribal regalia to be sold on the black market, things of that nature. So it varies from tribe to tribe.
This training was conducted by federal government contractor Wildlife Field Forensics. The definition of forensics is the use of scientific tests or techniques to detect a crime. The officers spent previous days in the classroom learning about the theories and techniques of forensics. A big part of forensics field work is the preservation of crime scenes, protecting evidence from contamination, and collecting clues such as DNA and footprints. Field instruction also focused on the investigation of a rare yet devastating event, a wildlife attack on a human. For that module, the instructor set up a scene with props and fake blood. Another instructor showed the officers how to extract ballistic evidence from a roadkill fox that had been shot post-mortem for instructional purposes. What most stood out in this field environment was the ever-present funk from the pair of carcasses enclosed by a chain-link fence. A decaying mule buck and a bull elk provided from government agencies from roadkill incidents, then kept in walk-in freezers for weeks to prepare for the course. They had spent the preceding 2 weeks decomposing at the site to set the scene of a poaching. This portion of the training was taught by Wildlife Field Forensics founder and director / former federal wildlife officer Carleen Goddard. When Carleen left the federal service, she became a student earning her master's degree in criminology and forensics from the University of Montana.
My background was working as a federal wildlife officer, law enforcement with U.S. Fish and Wildlife Service. And as a federal officer, working poaching cases, I developed a strong interest in time of death. At first, looking at carnivore time of death, but also interested in decomposition. And then when I got old, I went to graduate school and not doing government work anymore. And my graduate work was actually looking at decomposition over time to be able to do an estimate on time of death, because that's when we find poached carcasses, is long after the fact. And towards the end of my research, I thought of the idea, because when I was an active duty law enforcement officer, I looked for field investigation training, crime scene investigation training, specific to wildlife, and nothing existed. So I thought about the idea of, in 2007, thought about the idea of developing a program over a few days period, bringing in experts in their relative fields, doing wildlife crime scene investigation training. And that's how Wildlife Field Forensics was born. We did our first seminar in Montana, spring of 2007, and we've been doing it ever since.
Poaching, or the criminal take of wildlife, is a common type of crime that game wardens encounter. Usually it's the carcasses they find missing some key body parts, and the rest of the animal is left to rot. Game wardens and law enforcement rangers, when investigating a poaching case, can collect the insects feeding on a carcass, then send them to a lab where an entomologist or an insect scientist can learn the time of death based on the bug's development. Forensic entomology is the study of insects as evidence in a criminal investigation. In her research on analyzing decomposition to estimate time of death, Carlene also learned to collect and preserve insects for entomologists. Now she travels the country training conservation law enforcers to identify decomposition stages, collect and preserve insects, and how temperature and moisture affect decomposition and insect development. When it comes to insects, the main indicator of time of death are fly larvae, or maggots, as they're usually the first insects to feed on a corpse. With a group of officers going over their decomposition and analysis checklist handout, she asks the students to inventory the tools they need for the collection. This is where the story gets kind of gross, so you may want to stop listening if you don't want to go there.
Okay, so you've got your rubber bands, you've got your paper towels, you've got all your equipment. So what are you going to do? What's the first thing you're going to want to do? Glove up.
Okay, if you need help, let me know when we get up.
Throw up. Believe me, it's not that bad. And in the bloat stage, remember, that's the anaerobic. I mean, it's a balloon filled with gas. And once it's punctured by maggots or whatever, then it deflates, and then it goes into the active decay.
Both of these carcasses have already deflated, and maggots are pouring out from several orifices. They're pouring out collectively as a sort of liquid. The class breaks into groups and they begin to go for the maggots. Another piece of evidence they need to collect is temperature.
Okay, so I'm going to take the temperature. I'm just going to lay the probe on top and see they're pulling it down. They're pulling it down. I'm not pushing it. So that's the temperature of your maggot mass.
What is it?
And then you're going to do ambient. Okay, so we want to go ahead, grab a spoon, and
Look at that fried rice.
yeah, we're going to have fried rice for lunch.
Is that good or?
With gloved hands wielding a regular plastic spoon, they scoop the maggots into evidence cups, tapping the spoon to shed all the larvae. Then they soak them in hot water, which kills them.
Okay, then the next thing I'm going to do, they're dead, and that plumps them up, makes it easier for the entomologist to look at. Then I'm going to strain off the water. The hot water will plump them up and make it easier for the entomologist to be able to look at them, see the breathing spiracles on their butt end.
Then they put the plumped maggots into alcohol to preserve them. They prepare another sterile cup with live larvae, giving them some meat to feed on and enough air and room to survive the trip to the lab. In the process, they also record from which part of the carcass the maggots were collected. There are several stages of maggot development as well. Identifying the stages is important for an entomologist's lab work.
If it's got pink in the crop,
Yeah.
that's blood. It means it's still eating.
Oh.
So if it's a fairly large maggot and it's got a pink crop, it's probably second stage. If it does not have a pink crop, there's no coloration at all to the crop, and it's away from the carcass, that means it's done eating, and that's pre-pupal.
Could
And
they
that
be
would-
like these ones that are way away from the
Yeah,
body
you
there?
can see them moving away. I mean, they're already migrating off. And, you know, maybe some of these closer ones might still have a few with a pink crop, but these are probably all not going to have a pink crop. They're migrating away to pupate.
These maggots are young blowflies. They're the kind of flies with a green metallic sheen to them. They often show up minutes and sometimes seconds after something dies. They're very sensitive to the fumes from corpse and dung, as that's where their young thrive. But the blowfly isn't the only important insect indicator for time of death. Once rotten flesh is dried out or desiccated, it's difficult for maggots to eat it. This is where the beetles come in. Not the Fab Four, these carrion specialists come fashionably late to the feast. They're good at removing bits of meat from the bone. Some beetles are just there for the other bugs, predating on plentiful maggots and even on other beetles.
And then all those little beetles that are all around there too?
They're eating.
Like these little tiny beetles?
Oh, you know, I think the tiny beetles, I'm not an entomologist. I know what carrion beetle looks like. I've seen carrion beetle up here, beetle larvae. They're going to be eating, and the beetle that we see going in the ear,
Yes.
they're going to be eating eggs and larvae. You would collect those. Now, how are you going to collect your beetles? Let's have somebody doing a beetle collection here. So what are you going to do for the beetle collections? Alcohol, remember? Because you're going to put multiple beetles in there. You have one bottle that's going to be dedicated to your beetle collections. And that one bottle for all of your beetles, and you're going to be collecting the beetles all over the carcass. It's not site specific like your maggots are. You collect the beetles from various areas, you put them all in the same jar, and they're going to be put in alcohol because remember they eat each other.
Yeah.
And if you put a bunch of live beetles in there, all you're going to have left is one fat, happy beetle and legs of all the other ones.
Okay.
So,
Scooping up maggots and carrion beetles from the rancid putrefaction of decomposing flesh isn't a pleasant part of a law enforcement officer's job. However, the collection is just the beginning of the story for the forensic entomologist, whose results can shed light onto cases that would otherwise go unsolved. This field has been around since the 1930s and is used primarily by homicide detectives.
the one that we work through and use as a part of our training, she's an expert from Canada, forensic entomologist that has worked with Royal Canadian Mounted Police doing human homicide cases. More and more she's doing wildlife. So I'm kind of pinch hitting there and giving, training in collecting those insects and certain protocols that those forensic entomologists want those collections done under. They've got very strict protocols. And then those collections, those insects that are collected are sent to a bona fide forensic entomologist.
And how do they determine
The time of death?
yeah,
It's a complicated process and not so complicated. Temperature is what drives maggots. Maggots are the prime determiner that a forensic entomologist can determine time of death, even to a day, if they've got a good maggot sample and they've got good temperature history.
Maggots are pretty tolerant of cold temperatures. Some species have been observed surviving temperatures as low as 75 degrees below zero. In those frigid climes, they lie dormant in the carcass. When the mercury picks back up by changing seasons or night and day cycles, the larvae become more active. As their habitat gets closer to the upper 90s, they are able to eat and develop quicker. Determining an accurate outdoor temperature at the corpse takes a bit more field work.
We put a data logger out so we can get a range of temperatures over maybe a 2-week period, and then we look back at a nearby weather station, and those differences are constant, are consistent. So we can backtrack with the data that we have, placing a data logger out of poaching, and then we take that data that we've collected over maybe a 2-week period.
Thermometer data loggers are very accurate, inexpensive, and easy to use. They're basically a USB stick that the game warden leaves on the ground near the carcass site under a protective cover from the sun, with the east, west, and north ends open to allow for airflow. And then they can plug it into a computer and download the 1,000+ temperature readings that it records. This allows investigators to set up a sort of temporary weather station exactly where they found the carcass and to see how the temperature shifts overnight. Then they obtain the data logs from nearby public weather stations. They put those two data sets together and plugging them into an algorithm that compares the readings to pair the timestamps. This turns out a ratio or an average that makes it possible to go back in time, looking at the historic data from those established public weather stations. And from that, they have a scientifically accurate estimate of what the temperatures were around the carcass during the course of the decomposition.
Each species of maggot has its own development rate, and it's all driven by temperature. There's entomologists that have done nothing for decades but looking at different species of maggots. They're blowfly larvae. And under different temperature ranges, it takes this long for them to get to a certain stage in their development. There's basically 3 stages when they're still a maggot. There's first instar, second instar, third instar, and then they migrate off to pupate.
And while forensic entomology has been around for a while, it's still a very niche field. Carlene's program is one of only a few, if not the only one in the country, to teach this field collection technique to game wardens and law enforcement rangers. Insect collection in poaching cases isn't widely practiced by conservation law enforcement. Currently, the U.S. Fish and Wildlife Service has all of their new federal wildlife officers go through 2 days of instruction in wildlife forensics, contracted through Carlene's company. The service doesn't yet employ any forensic entomologists, so that's contracted out as well.
What I encourage people to do when they're at our training is to contact the woman that we use in Canada for our training, and she can tell them who in their area, who is a member of that international organization of forensic entomologists, that can give them, what would be the word, court credible evidence, or a determination that would be admissible as evidence.
That person from Canada is Gail S. Anderson, PhD. In 1997, 2 dead black bear cubs, along with their mother, were found in Manitoba at a dump site near Winnipeg. Their gallbladders removed. Bear bile, which comes from those gallbladders, is valuable in traditional Chinese medicine and demands a lucrative premium at market. Dr. Anderson had trained the area's Royal Canadian Mounted Police official who assisted the area conservation officer. They collected the insects from the dead cubs and sent the samples back to the lab. With Dr. Anderson's testimony on the time of death for those cubs, investigators were able to learn the identity of the perpetrators. Those poachers went to jail for their crimes.
When officers go to law enforcement academy, they get all the cop stuff. We get firearms training, we get defensive tactics training, all the case history training, but it's all geared to human type crimes. And once they finish their academies, whether they're a federal officer going to Federal Law Enforcement Training Center or a state academy, they're not getting wildlife specific training necessarily. We're kind of a novelty at this point, but we're still going. It shows that there's still a need. Every year at our May training in Montana, we'll have anywhere from 45 to 50 or more attendees from state, federal, tribal agencies. So there's a need. I mean, that to me demonstrates that there's a need. They're sending people. Poaching now, I hate to use the word poaching because that brings up the idea of hand slap misdemeanor. And a lot of prosecutors and even judges think back like 20 years of the hungry guy just trying to feed his family. And crime scene now, illegal take of a wildlife species is a crime, and people who commit it are criminals. And really upping the prosecutorial concept of what it is, that it is a serious crime, that's a state-by-state thing where I think it needs to be taken more seriously.
That was the founder of Wildlife Field Forensics and former federal wildlife officer Carleen Gonder for the U.S. Fish and Wildlife Service Southwest region. I'm Al Barris, and that's what's up.